So here we are on our mountain peak, and there's lots of directions in which we can look now that we understand DNA transcription and translation. And I particularly wanted to talk today about the immune system. I used to be able to spend more time in this class talking about human anatomy and systems of the body. Um, I don't so much now that we have, 1401 for students that need a course like this, but with more human biology content, and also because our school year used to be longer. Um, so I stripped all of that out, but it is timely to cover a bit about the human immune system. This is the way we defend against pathogens. And in case you forgot, a pathogen is any living or semi living thing that causes illness. Bacteria, certain protists, certain fungi, parasitic animals, and viruses. And we defend against those with our immune system. And if we happen to be born with no functioning immune system, we have to grow up inside a sterile plastic bubble. David Vetter was born in 1972 and had to grow up as, quote, the boy in the bubble, because with no immune defenses, he could have died horribly from a single cold. And understanding how the immune system works is critical, not just for treating patients like David Vetter, but for understanding things like vaccines, various other health concerns like autoimmune diseases and allergies. So hopefully this is one of the places where we can take all this abstract stuff I've covered about DNA and transcription and translation and all of that and put it into a context that you can apply. So, if we consider the immune system as everything that fights off pathogens, we can divide it into a couple of components. And one of these is the innate immune system. If you imagine the immune system as being like an army, defending against invaders, the innate immune system would be the barbed wire and the tank traps and the ditches and the minefields, because those work against any intruder. Those work against anything that comes into the body, and they don't have to be sensitized to it. So, for example, anatomical barriers to pathogens coming in the first place. Fever and inflammation are immune responses. A group of cells that actually can patrol virtually anywhere in the body. They're often called white blood cells, but they're not restricted to being in the blood. We call those, there's different types, but we most generally call those leukocytes. And finally, a group of specialized proteins called the complement system. So those anatomical barriers include things like your skin, flexible but tough and watertight and thick, and also constantly shedding its outermost layers. It does this a lot if you've gotten a sunburn, but your dead skin cells are flaking off all the time. You may not notice unless they build up and you end up with this kind of lizard skin effect on your elbows that you might have to scrub off in the shower. But because the skin is watertight, thick, tough, and shedding constantly, it's difficult for microscopic organisms to gain a foothold. I mean, occasionally they do. None of this is foolproof, but the skin is very good at protecting what's underneath it from bacteria that would love to eat it. Your stomach cranks out digestive enzymes and, of course, acid. Um, In addition to digesting your food, that breaks down most of the bacteria in your food. Occasionally, you can, of course, get food poisoning from bacteria that are resistant, especially bacteria that form endospores. If you remember that from way in the beginning, circa the third week, those may be able to get through, but most of them can't. Pathogens have a hard time getting into your eyes because your tears contain a powerful enzyme called lysozyme that breaks bacteria down. You digest bacteria with your tears. Sounded more poetic in my mind than when I said it. And just admitting somebody. The mucus in your respiratory system traps anything that you inhale, be it smoke particles or dust particles or bacteria and other pathogens. Those are trapped. The mucus is actually brought up and then ultimately coughed up. So potentially harmful pathogens can be swallowed or spit out on the sidewalk, although you really shouldn't do that before the bacteria have a chance to start growing in your lungs. Again, none of these is absolutely foolproof. People do get stomach infections and eye infections and lung infections and things like that. But they defend very effectively against the great majority of pathogens that are out there. There's the inflammation response, because whenever your cells take damage, they start sending out various signal molecules that trigger inflammation. Blood flow increases, leakiness of blood vessels increases, which causes fluid to leak out of the blood vessels, not whole blood, but plasma. And your nerves also become increasingly sensitive and you get pain. That's the four classic signs of inflammation, which sounds a lot better in Latin, by the way, because a Latin doctor named Galen wrote all of this down. He called it rubor et calor et tumor et dolor. That's redness, heat, swelling, and pain. And then fever overall is also an immune response. That heat, both the local heat of inflammation and then the total body heat of fever, sometimes that can interfere with certain pathogens. Bacteria that infect human bodies are often pretty finely tuned to live best at human body temperature, and raising the temperature even a couple of degrees can be enough to slow them down or knock them off their game, as it were. Fever also seems to enhance other immune defenses. Time was, anytime somebody got a fever, they might take drugs to try to bring it down. These days, that's no longer done so much unless the fever is getting high enough that it's starting to endanger the feverish person. If it gets over, I think, 103 Fahrenheit, 40-ish Celsius, I think. But yeah, fever is part of the immune response too. And then inflammation also triggers this population of white blood cells called leukocytes. Leuko is just Greek for white. And even though they're sometimes called white blood cells, they can patrol just about anywhere. They can slither right in between the cells that make up your blood vessels and move anywhere they're needed. Remember, when inflammation hits, blood vessels in the area become leakier. That causes fluid to leak out of the blood vessels and it causes the swelling of inflammation. It makes those tissues swell. That leakiness also lets the white blood cells crawl out and reach an infection site, and they can swallow bacteria or busted cells, broken up cell debris, or anything else they need to through a process called phagocytosis. I should mention phagane happens to be Greek for to eat, and cyto means a cell. So phagocytosis just means cell eating. And you can see a diagram of it down there below. A leukocyte will just crawl up to a little bacterium or whatever it is and extend some extensions of the cell called pseudopods and just wrap it around completely. And then once the bacterium has been completely engulfed, there's little bubbles inside the cell that contain this enzyme called lysozyme, which digests the bacteria. So a single leukocyte may kill and digest 100 bacteria in its life. And incidentally, if you have a bacterial infection, you can build up very large numbers of leukocytes, and that's what pus is. A mixture of dead and dying bacteria and active dead and dying leukocytes. Used to see a lot of those when I worked at the Berkeley Free Clinic. You probably don't particularly want to hear this, but what the hey, certain sexually transmitted diseases will cause pus to drip out of a man's penis. And we used to look at that under the microscope, and it was just loaded with leukocytes, fighting off the gonorrhea bacteria or whatever it might have been. And then finally, the complement system doesn't seem to be as widely known, but there's over 30 specialized proteins that play helper roles in the immune system. They can attract leukocytes. We don't need to go into exactly how they do it, but they can stimulate leukocytes to work harder, to swallow more bacteria or broken cells or whatever. And then there's some called the membrane attack complex that can literally punch holes in a pathogenic cell. So these things that are, you see cell membrane is diagrammed in blue and green. And these things that are labeled C5b, C6, C7, C8, and then a bunch of C9s, that's a series of proteins that can attach to the membrane of a pathogenic bacterium or protist or what have you, and literally blow a hole in it, which kills which kills whatever it is pretty effectively. Bacteria can't live with a hole through them. So all of that you can think of as being, static defenses that will work on absolutely anything. On, just like barbed wire or minefields will work on anything that crosses the area. Above and beyond that, we've got the adaptive immune system, and that is more like the cruise missiles of the body, because you can tailor the adaptive immune system to respond to very specific threats. Just like you can program a cruise missile to only strike a very precise location, one particular building, for example. The adaptive immune system can be tailored to respond to only specific pathogens. It's also capable of remembering specific pathogens for years. There's a phenomenon called immune system memory. And that means that the adaptive immune system can respond very quickly if it's ever invaded by the same pathogen a second time. This, by the way, is why certain diseases, if you get them once, you're not going to catch them again, at least not for a long time. So chickenpox and measles, for example, if you've had them once, you hopefully, sometimes it can fade, but at least for decades, you should have immunity to those diseases. And what we have to talk about here are two different types of white blood cell called T lymphocytes and B lymphocytes, and special proteins that B lymphocytes make called antibodies, which you've seen if you've ever accidentally kicked open an anthill. You've seen a bunch of little antibodies running around. That was a joke. Okay, so we talked about various types of white blood cell, and there are various types, different types of which um, may be produced by a body to deal with different types of infection. We don't need to go into here the difference between neutrophils and eosinophils and basophils and all of that. If you become a medical lab technician, you may end up having to do a differential blood count where you have to count different types of white blood cell in a sample, and those can tell you something about the type of pathogen that's invading. Anyway, there's another type of white blood cell called macrophages. Macro means big, and phagein is Greek for to eat. So a macrophage is a big eater. And there's yet another type of cells called dendritic cells. And the thing down below in purple that looks kind of like a carnation is a dendritic cell. What these cells will do is phagocytose whatever is triggering the immune system and pick up pieces of it and physically hand off pieces of whatever's triggering your immune system to cells called T lymphocytes. And the T lymphocyte here is shown in blue. I should mention that whatever is triggering your immune system is called an antigen. That's not the same thing as a pathogen. A pathogen is a bacterium, virus, or anything else that's causing infection. But you can have antigens that are not pathogens. A great example of this is allergies. If you're allergic to something, it means that your immune response is stimulated by it, even though it may not be making you sick, right? You know, peanut butter is not poisonous. You know, peanut butter is quite healthful and good for you, but there's a small fraction of people where if they eat peanut butter, the immune system immediately freaks out and goes into such overdrive that the immune reaction can actually cause far more damage than peanut butter ever could. And you go into this state called anaphylactic shock that I'm assuming you've heard of at some point. In that case, peanuts would be an antigen because they trigger the immune system, but they would not be a pathogen because peanuts do not make you, other than that, peanuts don't make you sick. Peanuts are not trying to grow in your body and take all of its resources for their own. Be kind of weird if they were. You'd have like peanut plants growing out of your ears or something like that. But that's why peanuts would be an antigen, but not a pathogen. An antigen is anything that stimulates the immune system, and macrophages and dendritic cells will pick up antigens. If it's an infection, they'll pick up pieces of the bacteria or pieces of the virus and hand them to T lymphocytes. Oh, T lymphocytes are mostly found in parts of your body called the lymphatic system. The lymphatic system is made up of some very thin little vessels that travel through your body. It's not, you could think of it as an extra circulatory system, except there's no pump. The lymphatic vessels don't have a heart pushing fluid through them, but your lymphatic vessels join little lumps of tissue called lymph nodes. If you've ever had a sore throat, the doctor probably felt your throat and your jaw for enlarged lymph nodes, these little lumps that you can feel when you've got a bacterial infection of your throat, like strep throat. If lymph nodes are enlarged in a part of the body, it means there's an infection in that part of the body. And the reason your lymph nodes enlarge is that that's where the T lymphocytes are found. And here's the thing, you are born with many millions of different types of T lymphocytes. And what makes each of those millions of lymphocyte types different is each one makes a differently shaped receptor protein on the surface. This is what a T lymphocyte receptor protein looks like. It actually sticks out of the surface like a little antenna on the surface of the cell. And you make tens, maybe hundreds of millions of different T lymphocyte receptors, but you don't have hundreds of millions of genes for them. What happens is that when these particular genes are transcribed and translated, portions of those genes are literally spliced and shuffled, which means that every one of your T lymphocytes at random produces a differently shaped receptor. So the macrophages and the dendritic cells go around searching for a T lymphocyte that has a receptor that's shaped in such a way that it matches the antigens that they're carrying around. So imagine a macrophage carrying around a piece of a dead bacterium, going up to each T lymphocyte and saying, hey, does this fit you? No? Okay, how about you? Does this fit you? No? Okay, how about you? Does this fit you? Yes! So eventually the right T lymphocyte is found. And that's what it looks like in this colorized computer graphic. The stuff up at the top and bottom of this picture that looks like, I don't know, like 2 layers of orange shag carpet, that's actually the cell membrane made up of molecules called phospholipids that I don't really have time to talk about. But that's the membrane of the macrophage up at the top and the T cell at the bottom. And here, I'll go to the pen. So this right here is the membrane of the macrophage or the dendritic cell. It's labeled APC, that just stands for antigen presenting cell. And right here is this antigen, this little fragment of a bacterial cell, maybe a bacterial protein that the APC is carrying around. And it's just stuck it onto a matching receptor right here, this blue thing on the T cell. So if the macrophage that's carrying around the antigen doesn't find a matching T cell, it'll go on to the next T cell. But once there's a match, that triggers the T cell to start dividing. And the T cell, as it divides, will produce 2 new types of T cells. One type is called cytotoxic T cells. They're also sometimes called killer T cells or killer Ts or CD8 plus cells. Those will go out through the body. They will kill virus infected cells. This is a major part of your body's defense against viruses. And by killing a virus infected cell, they can shut down virus replication before the virus can spread. They're also involved in the body's response to cancer. They will come up and try to kill cancer cells. And incidentally, because you were curious, yes, there are some therapies being tested that are trying to take T cells out of the body of a cancer patient and strengthen them and get them to grow and get them ready to go out and attack the patient's cancer cells even more vigorously. That's being tested right now. And I know this because my wife's mother was actually in a study that was being carried out at UALR, I might add, among other places. She was involved in a clinical trial of this, where they take some of her T cells, get them to grow in the lab, so she's got lots more of them, and jazz them up so they're ready to go after the cancer. My wife's mother has a type called non-Hodgkin's lymphoma. And in her case, it did work for a while. It did not provide a cure, and she had to go on chemotherapy after a while, but it did work, at least temporarily. So I'm sure research in this area will continue, trying to give cancer patients more killer T cells that can go after cancer cells. The other possibility is that these T cells will give rise to a second type, helper Ts, or CD4 cells. And I don't really have time to go into what those do, but let's just say they stimulate the immune response in various ways. HIV, human immunodeficiency virus, by the way, is hard to treat because it targets the CD4 cells. It's a virus that goes after the very system that's supposed to deal with virus infections. Kind of like crooks taking out the police station first. But there's another population of lymphocytes called B cells, and those can pick up antigens by themselves. If you have a bacterial infection or a virus infection, eventually broken up pieces of the bacteria, viruses, whatever it is, will reach a B cell. B cells are in your lymph nodes again, but some of them are constantly patrolling the body everywhere at any given time. And again, you have tens of millions of different B cell types, but you don't have tens of millions of genes for B cell types. Every one of those types of B cells has a different receptor protein. These little blue spiky things on the outside of the cell are B cell receptors. Every B cell has a slightly different shape of receptor protein. And what makes this possible is that as B cells are being formed in your bone marrow, if memory serves, the genes for those surface receptor proteins are cut up and shuffled. There's a neat little DNA rearrangement trick that means that of all of your B cells, everyone is different, or everyone comes from a different lineage, and every lineage makes a differently shaped receptor. So you have this huge diversity of cells crawling around in your body with random shaped receptors on them. And if a B cell encounters an antigen, if it doesn't fit, it'll pass it on. But if it does fit, if the shape of the antigen matches the shape of the B cell receptor, that B cell will start dividing repeatedly and turn into a type of cell called a plasma cell. Plasma cells are freaking protein factories. Plasma cells are like those World War II factories in America that were cranking out like one bomber every 90 minutes back at their height. Plasma cells produce ridiculously large amounts of antibody proteins, and this is what an antibody looks like. They're often diagrammed as looking like little letters Y, because they've got, kind of a stem right here, and then these forked parts there and there. Actually, that looks a little bit more like the Easter Bunny. See, there's the whiskers, right? It's the Easter Bunny. Okay, there. Okay, and those ears of the Easter Bunny Those are the parts of the antibody that are extremely variable. Every B cell has a particular shaped receptor that's created by this random shuffling of certain genes. And each B cell can produce antibodies that look like that B cell's receptors. So when a B cell finds an antigen that fits into its own receptors, that B cell starts cranking out antibodies that also will fit onto the antigens, physically fit onto them like a, key fitting a lock. You know, we use that metaphor a lot, but remember that the whole point of going into all of that god awful stuff about proteins that I did is that it's extremely important that proteins be made with the right shape. And this is one place where it's extremely important. Okay, what do the antibodies do? Well, because they have just the right shape, they will bind to antigens. They'll stick to them. They can actually cause the antigens to clump together into big lumps because antibodies will, one of those little bunny ears will stick to one bacterium or virus or something, and the other one can stick to a second one. So bacteria, viruses, etc., get entangled. Antibodies can block specific things. These are antibodies attacking a virus. And remember, these things that are sticking out right there, those are the spike proteins that the virus uses to recognize a host cell and attach to it. Well, the antibodies can get on top of the spike proteins and block the virus from attaching to a cell. Antibodies that are attacking bacteria will trigger the complement reaction. The complement reaction is always present, but the antibodies can make it stronger. And the antibodies also stimulate leukocytes, white blood cells, to engulf whatever they're bound to. That's a process called opsonization. And if memory serves, opson is the Greek word for to have a meal. You could think of the antibodies as making something more attractive and more tasty for those leukocytes to come in and engulf. You could think of the, I don't know, you remember the bit in The Lion King where Pumbaa is supposed to distract the hyenas by getting up and singing a song about how delicious he is. I can't actually quote the song, but he's forced to get up in a grass skirt and dance the hula and sing about what a tasty meal he would make. And then the hyenas come after them and, well, hilarity ensues. Kind of like that. You could think of the antibodies as kind of advertising. Hi there, Mr. White Blood Cell. Why don't you engulf this right here? It would make a really tasty meal. That's opsonization. You might recall, this was in the news, but there's an experimental COVID therapy where you can take antibodies from a person who has already had COVID. You can do this by taking some of that person's blood. If you're recovering from COVID, those antibodies are going to circulate in large numbers in the bloodstream for some time after you've had it. So you can take blood serum from a person who's had COVID, purify the antibodies, and then inject that into a sick person, give them extra antibodies that will attack their COVID virus particles. There's also ways in which you can make antibodies in the lab in various ways. There's a procedure called making monoclonal antibodies. This is actually, they've tried this for certain types of cancer. You can make antibodies to a certain type of cancer cell and then turn them loose by injecting them into the patient. This has also been tried for viruses, and this was done for President Trump. You might remember there was a lot of news coverage when Trump was uh, turned out to get COVID. So I'm checking the chat right here just to see if there's. Okay, great. Um, yeah, you might remember that Trump got this experimental therapy when he came down with COVID, and it did seem to work. This is what it was. They were pumping him full of pre-made antibodies to COVID. And in this diagram, those antibodies are these little things that look like Ys. And these arms right here are just the right shape that they can latch onto those spike proteins right here, interfere with the way they work, stop the virus from binding to cells in the patient's body. So that does work. But wouldn't it be great if you could make your own antibodies instead of having to get them injected into you before you even got infected? Well, have I got a deal for you? Some of both of those T cells that we talked about earlier and your B cells become memory cells. If your B cells are activated when you're infected with something, the plasma cells, some of those will basically put everything they've got into cranking out antibodies like there's no tomorrow, and they'll die pretty quickly. Others of them might last a little bit longer. But in addition to plasma cells, the memory cells may live for decades. This is a picture of a slice through intestinal tissues of a mouse. The blues and the greens are different types of cell making up the mouse intestine. And these red things, there's one and there's one, those are memory cells patrolling the tissues. And some of those might last a lifetime. And the upshot is if the body is ever hit with the same antigen, if you ever get the same virus or the same bacterium or whatever it is, those memory cells are already there, and they can very quickly divide and give rise to new cytotoxic and helper Ts and give rise to new plasma cells. Both T and B cells can become these memory cells. And it's those that enable your immune system to remember everything that it's ever faced before. It would be kind of like, I'm sure I'm sure every military does this, but the US military, keeps a lot of information on every weapon it has ever had to fight against before. So that if those weapons are ever used against the US military, they'll already know what the best countermeasures are. And I'm sure every military in the planet does something like that. Well, that's how your body can remember what it's faced before and fight it more efficiently the second time. Those memory cells, T and B memory cells. And incidentally, memory cells aren't just useful when you get infected by the same pathogen a second time. They also are used to produce more antibodies that go into breast milk. If a mother breastfeeds her baby, that baby's immune system is still not fully functional. Babies or newborns, especially, can be very vulnerable to infections because they are not quite up, their immune system is not quite strong enough to defend themselves. But mother's milk contains antibodies. It's loaded with antibodies to everything that the mother has ever been exposed to. So if mom has had chicken pox, antibodies against the chickenpox virus will be secreted in her milk. And for as long as that infant is breastfeeding, that infant will be protected against chickenpox, and so on for all of the other things that the mother might have had. And that's also the idea behind vaccination, is making use of those memory cells. If you challenge the immune system with something that looks like a pathogen but isn't, like a dead pathogen or a weakened pathogen or broken up pieces of a pathogen, fragments of viruses, bacteria, etc., or dead viruses, bacteria, etc., then the immune system can't tell the difference. The immune system will respond as if it were being invaded by live pathogens, by real viruses or real bacteria. And the memory cells will last for years afterwards, possibly for a lifetime. So when you get a vaccine, traditional vaccines, like, this doesn't apply to all of them, and not the COVID vaccines now. I'll talk about those in the next slide. But if you get a vaccine for smallpox or a vaccine against polio, or what's DTaP, diphtheria, tetanus, whooping cough, I think, DTaP, that's one of the ones that hopefully you got in childhood. Those are dead or weakened viruses, bacteria, etc. And your immune system responds to those as if you were really getting infected. So your immune system will fire up its T cells and fire up its B cells. The B cells will start cranking out antibodies. Incidentally, they can stimulate all of your immune system, which means why some vaccines make you feel sick for a day afterwards. You're not sick because you have a real infection. You're sick because the immune system is gearing up. So you might feel fever, inflammation, or things like that. Once that's all over, you'll retain the memory cells for many years afterwards, and possibly for a lifetime. You may have to get boosters, like you're supposed to get a tetanus vaccine once every 10 years, I think. There's some others that you may have to boost. The memory cells don't always last an entire lifetime. But now that you've got those memory cells, they're geared and ready to respond if the real pathogen ever attacks. So if you get injected with broken pieces of the tetanus bacterium, you'll be able to respond very quickly if the real tetanus bacterium attacks. I've gone a little bit over time, but I've got to show you this. People have tried injecting COVID virus proteins into the body, and that doesn't actually work that well. For some reason, those don't give you a strong immune response. But if you got Pfizer or Moderna, you got injected with messenger RNA, which codes for the virus protein, and that seems to work better. Your cells take that messenger RNA, transcribe and translate it. Your cells produce the virus protein, and then the virus protein activates your immune system. If you got the Johnson & Johnson vaccine, that's actually a crippled virus. You're getting a whole virus injected into you. That virus contains DNA that's transcribed and translated into coronavirus protein, and the coronavirus protein triggers your B cells. But the adenovirus, since it's been crippled, can't replicate in your cells. You don't get sick from the adenovirus. The adenovirus just delivers DNA that your cells transcribe and translate to make that coronavirus protein. And then your B cells crank out antibodies and create memory cells, and you're protected after that from coronavirus. And I'm sorry I went over time. I will go ahead and stop this now.